Alright, so this is the second half of your notes for this unit, and these notes are going to focus on viruses. So we've already talked briefly about why viruses are not alive. So here's a few more concrete pieces of evidence as to why. So they don't have organelles. So no organelles. They can't take in nutrients or energy if they don't have these structures to make that happen. They also cannot make proteins. Living things must be able to make proteins. And most important, they cannot reproduce on their own. They rely on a host cell. Kind of like a parasite almost. So, virus structure. Now, there's multiple shapes to viruses. In terms of this class in freshman biology, we're just going to focus on this main shape here. But here's another way that viruses can be structured. So, there's two major reasons to this shape, and it's called a bacteriophage. So you have the head part, which is this part, and then you have the tail. So the head has a capsid, and then the nucleic acid core, or your DNA. The capsid is just that hard outside covering that keeps it safe, and then the nucleic acid core is on the inside, all the squiggles. And then the tail is this part here, and then the tail fibers are these little guys. So he kind of looks like a giant robotic monster, basically. So a viral infection. For this to happen, a virus has to find a host cell. That's the most important part. You have to find a host cell. But it attaches to a specific site. So not every virus can attach and infect every cell. And typically, there's certain cells that are more susceptible, but it still has to have that specific site. And most importantly, most viruses cannot transfer between species. There's been multiple movies where that happens. It has happened in some rare circumstances, but that's like if your dog's sick, you're not going to get sick too because it can't transfer between species. So types of viruses, like I said, the bacteriophage is what we're going to focus on. So it's a virus that infects bacteria. That's something you definitely need to know, and it's in the name, so that shouldn't be too hard to remember. And you have to know the structure. Expect to label the structure. So it's labeled over here, and then you'll have an unlabeled picture on a quiz or a test. So over here, the head or the capsule is this big outside covering. So we did that. The tail is this royal blue part, and it's kind of like a syringe. It's what injects that DNA into the unsuspecting bacteria cells. The tail fibers are the little leg-looking things. And then the DNA is not in this picture over here, but it would be just squiggles on the inside. So the bacteriophage life cycle. So there's 6 major steps. I'm not going to make you memorize the specific names of each step, but you should be able to summarize it in simple terms as far as what happens. So you have your virus. It's going to come inject your bacteria cell, which is this oval. The nucleoid that you see right here, that is like saying it's the DNA of the bacteria. So what happens is we go on to step 2. So the DNA from the virus is now inside the bacteria, but it doesn't connect with the DNA of the bacteria. It's just on its own. But the problem is this DNA from the virus is going to destroy the cell, because that DNA uses the inside of that cell as a prime breeding ground to produce more of itself. So now you have all these little DNA pieces of what was in the virus. So now they start to assemble, they start to form all the parts that they need. And then they actually assemble. And then what happens is the cell bursts. That's how these escape. So the cell bursts, and all of the new viruses escape into your body to go infect more cells. So the basic steps, this is in written form. So you have attachment to the host cell. Then you have the release of the viral genes, destruction of the host cell DNA. It ruins it, and it also replicates viral DNA. Okay, then that's that step 4. Assembly of the components, and then the release of the particles. Really important to note here that the host cell is destroyed. So now we have two different cycles that these cells can go through. So you have the lytic and the lysogenic. A really easy way to keep this straight is lytic is shorter than lysogenic in terms of letters in the word. So lytic is a shorter cycle. And lysogenic is a longer, or technically lifetime. Well, lifetime of the organism. So let's look at just the lytic cycle. So first of all, it occurs separate from the host cell DNA in terms of the replication. And then copies are made and sent back to the environment. Like I said, it's a short-term disease, such as the common cold, influenza. Lysogenic, on the other hand, is a longer, and the word's longer, so it occurs within the host cell DNA. That's the most important difference right there. You need to know the difference between this and this. And then as a result of this first bullet, the viral DNA becomes part of the cell's DNA. These are diseases that you can never get rid of. They are always in your body because it's part of your cell's DNA. So your cells are never going to all die off. So you're always going to have some of this virus in your DNA. It's a long-term disease. And when you go into outbreaks, such as like if someone has a herpes outbreak, the DNA that was in the cell goes out on its own and then goes and infects cells like the lytic cycle does. So that's why people with AIDS or herpes tend to have outbreaks, or they have some good days and bad days, because it's very unstable. The cells are very unstable that have this DNA. So here's a closer look at the lytic cycle. So if we start up here, you have your virus. It's going into the cell. This looks a lot like the typical bacteriophage replication. And then this host cell is destroyed. I keep focusing on that because that's the most important thing. The host cell is destroyed at the end of the cycle. The lysogenic cycle, on the other hand, the DNA inserts or integrates into the host cell. It becomes part of the nucleus, the most important part of this one. If you look at this picture closely, you see this little dark blue strand. It eventually becomes part of the DNA of the cell. So it's permanently there. It will always, always be there until the organism dies. So let's look at advantages and disadvantages. So lytic cycle viruses, the advantage, it's fast reproduction. This is why when you get a cold or the flu, it hits so fast because these viruses are just continually attacking, and then they just produce more and more and more. The disadvantage, back to this host cell again, the host cell is killed. So DNA cannot be passed on. It's kind of like back to the beginning of the year talking about fitness of an individual. So the host cell is killed, so the DNA is not passed, and as a result, the disease really doesn't continue on. That's why a cold or a flu is a short-term thing. Lysogenic, on the other hand, the advantage is the viral DNA is passed on, so it's the opposite of the lytic cycle. The disadvantage is the replication takes longer because it uses the host cell's replication schedule, not its own. So basically, one's advantage is the other's disadvantage. So that's an easy way to keep that straight. And then here's just another diagram for you. So this part here is the lytic cycle. And then this down here is the lysogenic cycle. But as you can see, the virus can leave. It's part of the cell here, the red part, but it can leave and then it goes out on its own to infect a cell and enter the lytic cycle, which is where those outbreaks come in. And then last up is retroviruses. So viruses are retro if they have RNA. An easy way to remember this, RNA starts with R, retroviruses starts with R. So they have RNA, but what do we know about DNA? You need DNA to do anything for the cell and to code for anything important, and DNA is in the nucleus. So if a virus has RNA, it has to figure out a way to get that RNA into DNA. So what it does is it uses an enzyme called reverse transcriptase. This enzyme changes RNA into DNA. And then, guess what? That host cell's DNA is then going to infect the virus's DNA is then going to infect the host cell's DNA. And this, the most common form of this is like HIV and AIDS. It's considered a retrovirus. Some cancers are also retroviruses, such as leukemia, more specifically T cell, in case you're curious. But that is the end of your virus notes.